This is Module 3, Oxygen-Binding Proteins and Methods for Analyzing Proteins. In this video about myoglobin, I'm going to talk a little bit about why we need oxygen-binding proteins, and specifically about how myoglobin is used to store oxygen in body tissue. So why do we need oxygen-binding proteins? There are several reasons, actually. So first, oxygen is critical for cellular respiration and survival. Remember that oxygen is a required reactant for the process of cellular respiration. So it is the last electron acceptor in the electron transport chain. And so oxygen is required for cells to metabolize sugar to create ATP. Oxygen is also poorly soluble in aqueous solutions, which means when oxygen enters the lungs, and it moves from the airspace into the bloodstream, it's not actually very soluble at all in the bloodstream, and so it would be difficult to carry oxygen to all of the tissues that require it for this reason. Oxygen also cannot diffuse over great distances, and so because oxygen enters our bodies at the area of the lungs, it would be very difficult for oxygen to naturally diffuse to different areas of the body without the aid of the bloodstream. Even with the aid of the bloodstream, when oxygen finally is delivered to the tissues that require it, it has a difficult time moving from the bloodstream and from capillary beds and to permeate all of the tissues that need it in order to metabolize sugar and fats. And then lastly, we need oxygen binding proteins for distribution of oxygen. So we need to find some way to make it soluble in the bloodstream and then distribute it to all of the tissues and cells that require it. And we also need to be able to store oxygen. This is more related to how your muscles utilize oxygen and do cellular respiration when they're active. When your muscles become active, so say you just start going for a run or something, or you start exercising, your cells will use up all of the available oxygen in your tissues, and so very quickly. And so we need to find ways to store oxygen in your tissues so that you have a little bit more of a buffer region. So like a little bit more oxygen is stored in your tissues so that you can access it during periods of activity. So there are two different oxygen binding proteins of critical importance in our body, myoglobin and hemoglobin. And for the rest of this video, I'm going to talk about myoglobin, which is the primary oxygen storage pigment of muscle tissues. So its primary goal is storing oxygen, not transporting oxygen. And the structure of myoglobin itself enhances its ability to do that. So let's take a little bit of a closer look at the structure of myoglobin. Myoglobin is a globin protein, so it's a globule protein. So it's spherical, roughly spherical in shape, and has the globin fold motif, which means it's a polypeptide with several alpha helices clustering together in a tertiary structure. Specifically, there's 8 alpha helices. They're labeled here, A through H on this diagram, that cluster together. And then there's this single heme group in the center, which is considered to be something called a prosthetic group. It's not necessarily protein in nature. It's a different type of molecule, a different compound, mostly a slightly nonpolar compound, so as to dissolve oxygen a little bit better in the bloodstream. So it binds nonpolar substances fairly easily. So it itself is also nonpolar. It's found in the muscle tissue of all mammals. This protein, myoglobin, is found in muscle tissue, mostly because its role is for oxygen storage. And so it helps to, it remains a readily accessible form of oxygen for muscle cells to use when doing high rates of cellular respiration. It also facilitates oxygen diffusion in muscle tissue, so it allows for oxygen to be distributed from any capillaries through the entire length and depth of the muscle tissue. So it carries it from the bloodstream into the muscle tissue and then stores it there. It also increases oxygen solubility in muscle cells. So muscle cells are surrounded and bathed in interstitial fluid, much like every other tissue in your body, is surrounded and bathed in interstitial fluid, which is an aqueous environment. And so for oxygen to diffuse through muscle tissue, it must be able to move through the polar interstitial fluid. Since oxygen has a difficult time with this, binding first to myoglobin makes it easier for oxygen to move through the depth and length of a muscle tissue. Let's talk a little bit more about that heme group that I mentioned. So this is a prosthetic group. It consists of multiple porphyrin rings with a single ferrous ion contained in the center. So this iron atom has 6 possible bonds. 4 are bonded to these nitrogen atoms on the porphyrin ring, and that gives it some kind of structure. It gives it a place for it to, like a scaffolding for it to sit in. And this is actually the heme group right here. 1 is bonded to a histidine residue of the rest of the protein. And so this anchors the prosthetic group in the rest of the protein that makes up the myoglobin. And so this is a separate group, but it becomes attached to the rest of this protein. So you can sort of see its attachment here via this histidine residue. And then another one is reversibly bonded to oxygen. So it's bonded to oxygen when it's storing oxygen, and then it's got one extra bond, iron is when oxygen is not bound to it, it's open and ready to bind oxygen again. When heme is contained within the protein or buried towards the center of the protein, iron cannot be oxidized and it stays as free iron. Iron only binds oxygen in the Fe2+ state, and so in this state. So when oxygen becomes bound to it, it cannot bind any other further molecules. And so how much oxygen is bound to myoglobin? A single molecule of oxygen, O2, is bound to a single myoglobin protein. How, then, can we measure this? So we can measure the interactions between a protein and its ligand, which is, so for a ligand is essentially anything that will bind to a protein's binding site. If it's an enzyme, the ligand would be the enzyme substrate. If this protein is a receptor, the ligand would be the hormone. In this case, in myoglobin, the ligand is oxygen. And so we can measure ligand-receptor interactions through these special types of analyses called binding curves. Ligand binding is referred to in terms of the P50. And so the P50 means so the P50 is defined as the concentration of the ligand that binds 50% of the available receptors. And so to transform that into myoglobin, the P50 would be the concentration of oxygen required to bind 50% of myoglobin proteins in solution. This is actually referring these other characteristics, a higher affinity enzyme and a lower affinity enzyme, is referring to specifically how an enzyme binds its substrate, and which is measured using the same kind of analysis with these binding curves. And so all I'm trying to say here is you can actually measure the affinity for which a ligand will bind its receptor by looking at its P50. So let me try and explain what this means in the context of an enzyme. So here we have 2 different enzymes in one substrate concentration. So we have one substrate, 2 different enzymes that are both capable of binding that substrate, but to various degrees. Let's look at the P50, which is the 50% binding. If we move over here, we see that it takes only about 10 nanomoles of substrate to occupy 50% of the binding sites of the enzymes in the solution for this enzyme. And so because it doesn't take a lot of substrate to bind half of the available enzymes, we would consider this to be a high affinity enzyme. It binds its substrate very well, tightly and quickly. If we look at this green line here, what we're seeing is that 50%, it takes about 30 nanomoles of substrate to bind 50% of these available enzymes. And so you need to basically overload the system with lots and lots of substrate just to encourage the enzyme to bind it. So let's take a look at this when specifically the ligand is oxygen. Um, so the receptor protein here is myoglobin. Oh, that's a spelling error. Sorry about that. I can actually fix that right now. Sorry about that. So the receptor protein is myoglobin, also referred to as M lowercase b. And then in other curves in the next video, we'll see how we adjust this for when the receptor protein is hemoglobin, or H lowercase b. This, because it's oxygen is the ligand, we specifically call this an oxygen binding curve. Theta right here is the fractional saturation, or basically it's equivalent to the P50 in an enzyme binding curve. And so what this um, is telling you is basically the same thing as P50. So it's a measurement of ligand concentration when 50% of the receptors are bound to ligand. So again, transforming that into myoglobin, it would be representative of the substrate concentration at which 50% of the myoglobins in solution are bound to oxygen. When myoglobin binds to oxygen, it does so in a hyperbolic curve. So basically, as oxygen increases, and so that's going to be this on here on the x-axis, as oxygen concentration increases, more oxygen is bound to myoglobin until all of the myoglobin is saturated, and it does so at a very fast rate. So you can see the slope here is very steep. And so what this is telling us is that myoglobin binds oxygen with very high affinity. Let's consider changing different concentrations of oxygen to kind of, well, first actually, let's look at the P50 or the theta value. And so what this is telling us is that the point at which myoglobin is half saturated is only 0.4 kilopascals of oxygens, which is a measurement of oxygen concentration. Yeah. So 50%, so this is a really low number. So it doesn't take a lot of oxygen to half saturate all of the myoglobins in solution. Let's take a look at the oxygen concentrations of the lungs and the tissues. When you look at the oxygen concentration of the lung, it's actually not even on this scale. The amount of oxygen in the lungs is so high that almost immediately myoglobin would be saturated. However, you don't find myoglobin in the lung, so that's not really totally relevant. Where you do find myoglobin is in the tissue, where oxygen concentration is about 3 kilopascals. So that would be around here. And you can see that in the tissue, most of the myoglobin available for binding oxygen will be saturated with oxygen. So again, 3 would be about here, and that represents about 90% of the available myoglobins would be expected to be bound to oxygen. Therefore, under normal physiological conditions, myoglobin is almost totally saturated in your body tissue. Okay. When talking more about P50, and so this basically means that the affinity of myoglobin to bind oxygen is very high. The lower the P50, or the lower the theta value, or actually the theta value is really referring to the percent of the receptors that are bound. We're still talking about P50, which is actually a substrate concentration. So I just want to make sure that that's clear. P50 is a substrate concentration at which half of the myoglobin is saturated, and the theta value is the percent of saturation. Okay. So again, P50 would measure the affinity of myoglobin for oxygen. If that number is very low, that means less oxygen is required to bind 50% of the myoglobin. So you don't need a lot of oxygen around for the myoglobins to be able to bind it and hold on to it. If there was a high P50, this would mean that more oxygen is required to bind 50% of that myoglobin. So a high P50 means low affinity. A low P50 means high affinity. Because myoglobin has a very low P50, that means it has a very high affinity for binding oxygen. And so whenever oxygen is present in the tissues, myoglobin will grab it up very quickly and hang on to it. And most of the myoglobins in your tissue remain saturated with oxygen until they release it for use by your cells for cellular respiration.